Hello friends, in this video lecture let us talk about a new topic called acid-fast bacteria. Certain gram-positive bacteria possess a characteristic called acid fastness, according to which if a bacteria is stained with a basic dye and then washed with acid solutions, it retains the basic dye, it does not lose it. Other bacteria, those which are not acid fast, they tend to lose that stain on acid wash. So basically acid fastness is such kind of characteristics, that their hunger for the acid. So to show that a bacteria is an acid fast, one has to do a kind of staining technique that is called Gill-Nielsen staining. In this procedure, you have to make, as usual, a smear of the bacterium on a slide. Say a drop of bacterium is taken, bacterial sample, and that is dragged with another slide to make a smear of the bacterium. Thin film of that. So this is a smear of the bacterium. After that is heat fixed, like other staining procedures, and then it is followed by treatment with phenol. Phenol containing some basic dye like the carbofuchsin. It is a red dye that exhibits red color. So after treating with phenol containing basic dye, cells are washed with water and then they are washed with acid, mineral acid, 20% solution of hydrochloric acid or sulfuric acid. The acid-fast bacteria, you can divide here in two groups: the acid-fast and non-acid fast. And the non-acid fast. The acid-fast bacteria will retain the color; they will not lose it. They will be red under the microscope, but they will lose the color and they will be unstained or destained. Okay, so this attribute of the bacteria, this characteristic of not losing the basic stain on acid wash, is called acid fastness. This is shown by a group of bacteria called Mycobacterium. Mycobacteria in plural. The Mycobacterium tuberculosis, Mycobacterium smegmatis are the example of that. This is non-pathogenic form of Mycobacteria. So these bacteria exhibit such characteristic because they contain a kind of a chemical wax that has been called mycolic acid. So they contain mycolic acid in their cell wall, which is basically similar to those of gram-positive bacteria. That is, they contain the peptidoglycan. So if this is plasma membrane and this is the peptidoglycan layer, thick peptidoglycan layer, just like the other gram-positive bacteria, they contain within this peptidoglycan layer, they contain certain kind of organic molecules, large organic molecules. Those are waxes, and they have been called mycolic acids. These mycolic acids, they hold the stains, the carbofuchsin basic stains. They combine with these mycolic acids and cannot be expelled out. That's why these acid-fast bacteria, they retain the basic dyes. As far as the structure of these mycolic acids are concerned, they are very complex structures. In case of Mycobacterium smegmatis, the structure is like this. It is a long chain of hydrocarbon like this. CH2 1 minute. Good night. Then there is a double bond here. Then again 13 carbon length molecule. Again there will be a double bond unsaturation. followed by still it is continuous with 17 carbon stretch, and then again there is a double bond, and then the beta hydroxyl group over here. This is the alpha carbon, and this is the COOH carboxyl group where the chain ends. In this alpha carbon, there is a long chain hydrocarbon again. So this is the structure of mycolic acid that is present inside the cell wall, that combines with these toxin molecules and retains the color even after acid was. So they are acid resistant. People have extracted these and treated them with acid. They do not lose their characteristic. So this is the reason this mycolic acid has been attributed for the characteristic of these bacteria, this acid fastness nature of this bacteria. That's all about the acid fast bacteria. Thanks for your attention.